Okay, hello. My name is Carlos Cavallaro. I am a sophomore here at Oglethorpe University. I'm a biology major, and this is my presentation on tetrahedral framework nucleic acids and alleviating insulin resistance. So first of all, I want to start off with a general outline of the presentation and what I'm going to be telling you guys about. So first of all, I'm going to start off with an introduction on the topic, and I'm going to talk about my general personal connection with the topic, why I chose it, why I'm interested in it. Then I'm going to get into some background information on the different themes that I'm going to delve into, explain the details of those, and talk about the greater themes of this specific topic. After that, I'm going to get into the details of the study that I chose to analyze and talk about within my presentation, go through the different experimental questions that were asked, the hypothesis that was made, and the general conclusions of that study. And then at the end, I'm just going to talk about some concluding points, talk about some general take-home ideas of my presentation, and then my works cited page will be at the end. So starting off with the introduction. So just as a basis, I want to start off by talking about the relationship between insulin and glucose and how that ties into my topic. So as we all know, insulin is a very important molecule within our body that we need in order to take in glucose into our cells so that that glucose can be converted into ATP in the form of energy that our body needs to function. And so essentially what insulin does, it's secreted by the pancreas, and what it does is it binds to insulin receptor sites on the human cells, and it basically allows then for the glucose that's in the bloodstream from different things that we consume to then enter into the cell. And so, as I said, insulin is very important, and we really need it in order to properly function. And so therefore, then going into my general topic about insulin resistance, insulin resistance is really harmful to the body, because essentially what happens is these cells are not going to be able to respond as well, and they don't have as many functional insulin receptor sites. So therefore the insulin is not really able to do its job as well in letting the glucose into the cell. As you can kind of see on this figure on the right, at the top is the normal cell. All the insulin receptors are working properly. The insulin is able to bond, and then the glucose enters the cell. Whereas on the bottom, in the more insulin resistant cell, some of the receptors are not functional, and therefore the insulin is not able to bond, and very little or not as much glucose is entering the cell. And so basically insulin resistance often can occur in organisms that are affected by metabolic syndrome and metabolic diseases, which I will go into a little bit later in the presentation. But insulin resistance overall is really harmful to the body, and is something that we want to be able to treat. And like I said here, if we are able to treat this insulin resistance, it can help to alleviate and reverse these effects of said metabolic diseases, as it will help to allow more glucose to enter the cell, bring blood sugar levels back to a more normal range, and allow the body to function more properly. So moving on, I want to talk about diabetes and my kind of personal connection to this presentation and this topic. So I myself actually have type 1 diabetes, and I've had it for about, about 5 years now. So yeah, I was interested in looking at things that relate to diabetes and the cure and the treatment of them. But I'm going to go ahead and go into the differences between type 1 and type 2 as they are pretty different. They have different mechanisms and different reasons, although they do share similarities. Obviously, they both fall under the umbrella of diabetes. So for type 1, which is what I have, the pancreas simply does not produce any insulin at all. And therefore, if there is no insulin in the bloodstream, there is no way for the glucose to naturally get into the cells whatsoever, regardless of how functional these receptors are. So in type 1, there is just no insulin present, and therefore these individuals, including myself, need to take in insulin in the form of a shot or a insulin pump into their body, and that way they can take the glucose into their cells. Whereas for type 2, as I was talking about earlier, these cells are just not as receptive to the insulin. They don't respond to the insulin as well. And so therefore there is still insulin being made and it's still present within the body. However, the cells are just not absorbing the glucose well because of this insulin resistance. And again, as I've been saying, insulin resistance is one of the major players within type 2 diabetes. And so if we're able to alleviate this resistance, it can help to treat and potentially fully cure type 2 diabetes, along with other steps such as healthy diet and exercise. There is also studies being done on how these tFNA's can help to also reverse new onset type 1 diabetes. This is a whole different topic that I'm not going to go into as much detail upon. However, it is a potential, it does have some potential in this regard to type 1 diabetes. However, the mechanisms to treat can definitely be much, or would definitely be much different than the mechanisms at stake here in treating type 2. So going on to metabolic syndrome and obesity in general. So what metabolic syndrome is essentially it's a cluster of different conditions that include increased blood pressure, higher blood sugar levels, overall obesity, and then just abnormal cholesterol and triglyceride levels. And essentially, these are all things that kind of tie back in with insulin resistance. And essentially, I took a quote from the study that I will go on to discuss that said obesity-induced insulin resistance is the hallmark of metabolic syndrome, and chronic low-grade inflammation links obesity to insulin resistance through the activation of tissue-infiltrating immune cells. So all of these tie in together, and all of these conditions have will cause the increased risk of more severe diseases such as diseases of the heart, stroke, and again, type 2 diabetes. So now moving on into the mechanisms of what is talked about in the study as helping to combat this insulin resistance and these metabolic diseases, and that being tetrahedral framework nucleic acids. So many of you might be asking, well, what exactly are these tFNA's or tetrahedral framework nucleic acids? They're actually very prominently used and complex, but very interesting. Essentially, they're 3D DNA nanomaterials that are man-made, that scientists make in a lab out of nucleic acid subunits. And so they are made, and as I said, they're widely utilized in the biomedical field. And essentially their main use is as a sort of vehicle to help transport and deliver certain functional molecules or drugs into the body, into the bloodstream, wherever the goal is. And essentially what they do is they can take these drugs and incorporate the drug into these tFNA's, and then deliver this combination of the tFNA and the drug into the body in order to help more efficiently deliver the drug, make the drug more effective within the body. And specifically in this case, in this study, the drug that they chose to try and make more effective is resveratrol. And so I will talk about this more in depth on the next slide about what resveratrol is, and then go into the study itself. But essentially they wanted to take this resveratrol, incorporate it in with the tFNA's, and then deliver it into the body in hopes to make the resveratrol more effective and in doing what they wanted it to do. So what resveratrol is it's a natural chemical compound. It's found all over the place in nature, in any grape products, peanuts, cacao beans, all types of different berries. And basically it's it's used as a supplement. You can find it in pharmacies. And basically it's known as an antioxidant and anti-inflammatory. It's known to help overall bodily health. It helps with, as I said, reducing inflammation, promoting organ health, and it can help to protect against cancer, diabetes, Alzheimer's, diseases such as this, and also overall just help with increasing insulin sensitivity, increasing the effectiveness of insulin within the body. And basically, it's a good example of a drug that can be incorporated into these tFNA's, as I was talking about, and this combination of the drug and the tFNA can then be delivered. And as I was saying, in this specific study, the goal was to incorporate resveratrol into the tFNA to help improve, excuse me, the effectiveness of the resveratrol in alleviating insulin resistance. So moving on, so just a little intro into the study that I'll be talking about. So the name of the study is Tetrahedral Framework Nucleic Acid-Based Delivery of Resveratrol Alleviates Insulin Resistance from Innate to Adaptive Immunity. So basically, going into the experimental question, so what they said, what they started out with saying was that current therapeutic approaches lack efficacy and immunomodularity capacity. And what they're talking about here is therapeutic approaches of drugs such as resveratrol in helping with diseases such as type 2 diabetes and other metabolic diseases. And so basically they said thus, in this case, a new therapeutic approach is needed to help prevent chronic inflammation and alleviate insulin resistance. So what they wanted to ask in the study was, will the tFNA and RSV combination, essentially what from here on out, what I'll be calling the molecule that is the combination of the tFNA and the RSV is tFNA-RSV. So will tFNA RSV help to alleviate insulin resistance? That was their question. And they went ahead and said that their hypothesis was that the combination of tFNA's and RSV will improve the properties and therapeutic efficacy of RSV and enhance the immunomodularity capacity of the tFNA's, as well as improving insulin sensitivity by promoting M2 macrophage polarization and stabilizing the T cell ratio. So essentially their hypothesis was, yes, this combination of the drug and the tFNA will help alleviate insulin resistance. And that was their overall goal. They wanted to prove that the tFNA's are a great way to deliver a drug, in this case, resveratrol, that will help to combat insulin resistance. And eventually down the line, hopefully help to treat diabetes and other diseases and things that come with obesity and such in humans potentially. So moving on, so getting into the materials and methods. So basically what they started out by doing, they took 4 single-stranded DNA strands, and they wanted to, out of these 4 strands, they wanted to make this tFNA nanomaterial. So what they did was they took these 4 strands, they added in a HCl and MgCl2 buffer, at an equivalent molar ratio. And they heated up these strands to 95 degrees Celsius for 10 minutes and cooled them to 4 degrees Celsius for 20 minutes. So basically, once they combined these strands and heated them up, did a heating and cooling process, this then created the tFNA material. And so then after that, what they wanted to do was incorporate the RSV, or the resveratrol. And so as you can see on the figure here, they had the self-assembly of the tFNA. And then in order to incorporate the resveratrol, what they did was they took different concentrations of the drug, up 20, 40, 80, 120, and 160 micromolar. And they added these concentrations of the drug to the tFNA solution that they had just created. And they stirred the mixture for 6 hours at 4 degrees Celsius. And then they went ahead and took out the residual single-stranded DNA and RSV using ultrafiltration. So after all this, they had the combined tFNA RSV molecule. And so then they wanted to get mice to use for their experiment. And so they obtained male mice that weighed on average 20 grams. And what they did was they started out with all of the mice together. They did a week of adjustable feeding. And then after this first initial week, they took the mice and randomly divided them into 2 separate groups. So in this figure, it only shows the high fat diet fed mice, but as I will explain, there was also a control group that was fed a normal diet. So they split the mice into 2 groups. One group was the normal diet, which is not shown on this figure. And then the other group was the high fat fed diet. So the normal diet was fed a normal chow diet, whereas the high fat diet group was fed a diet that consisted of 60% calories from fat, 20% calories from protein, and 20% calories from carbohydrate. And so after 8 weeks of feeding this diet to the high fat fed diet and then the normal fed diet, after 8 weeks of these, the mice in the high fat diet were then split up into a further 2 groups. And one group was injected with normal saline as a sort of control. And then the other group was actually injected with the tFNA solution that they had made prior to the start of these diets. So the normal diet group was, as I said before, used as the control. And then after this, all groups were administered equivalent drug volumes for 6 weeks, whether it was a saline or the actual tFNA solution. So after the 6 weeks of treatment, they went ahead and killed the mice and extracted their liver, fat tissues, and skeletal muscles, and performed 2 different types of tests on them. One was called an intraperitoneal glucose tolerance test, and the other one was called an intraperitoneal insulin tolerance test. And so this test was performed on the conscious animals, so the glucose tolerance test was performed on the animals after they did a 12-hour fast, followed by an injection of 2 grams per kilogram of body mass of glucose. And then for the insulin tolerance test, they went ahead and did a 4-hour fast of the mice, followed by 0.7 units of regular human insulin per kilogram of body mass. And so then after performing these tests, they took the blood glucose, by taking the blood glucose levels from the tail vein of each of the mice. This is what allowed them to do the test. So that was the main materials and methods. So moving on to the results. So at the top is kind of, it's a separate figure. It's not aligned with the figures below it. It's kind of its own figure, just showing again a picture of kind of the general timeline of I'll move my picture here. The general timeline of how the experiment was performed. So at 0 weeks, they had the mice. They did the high fat diet for 8 weeks. And then at the 8-week mark, they stopped the diets, and then they went ahead, they continued the diets, but they went ahead and did the treatment after the 8 weeks for another 6 weeks and saw how the mice responded to that treatment. So as far as the results go, so part B of this figure just shows their overall glucose levels in the bloodstream. And actually, before I get into talking about the figures, I just want to point out to you guys at the bottom here, it shows the different groups of mice that correspond with the different numbers on the figures. So group 1 was the control group that was fed the normal diet. Group 2 was the group that was fed just the high-fat diet with no treatment. Group 3 was the group that had the high-fat diet plus only the resveratrol and nothing else. Group 4 was the high-fat diet plus only the tFNA's without the resveratrol. And finally, group 5 was the group that had the high-fat diet plus the tFNA RSV combination treatment. So as you can see, group 5 is the one we'll mainly want to be focusing on. And so for part B of the figure, you can see how overall their group 5 had group 5 had a much lower glucose level and much more comparable to the control group, or essentially normal mice. Part D of the figure, or sorry, I'll start with part C. Part C again showed the weight, showing that group 5 again had a weighed much less on average than the other 3 groups that had the high fat diet, and was much more comparable with the control group. Going into D and F, these two were the prior glucose sensitivity and insulin sensitivity tests that I was talking about. And essentially what they're testing here is just the response to the insulin and how efficiently the mice were able to use the insulin and take in the glucose. And as you can see, group 5 was usually the quickest and most efficient in keeping their blood glucose levels low and returning them back to normal. And so, yeah, so that's the main part of that figure. So moving on, or just one more point about that last slide. Basically, what we can see here is that the tFNA RSV infusion did help to improve insulin sensitivity in that group 5 of mice. So moving on, this talks about the macrophage polarization in the adipose tissue, which is the fat tissue of the mice. So essentially what's been found in previous studies is that macrophages, the accumulation of them in the adipose fatty tissue has been identified as the source of pro-inflammation and essentially just the development of obesity-induced insulin resistance. And so also in a previous study, it's been shown that the expression of the TNF-A gene shows increased obese adipose tissue, and that neutralizing this TNF-A gene will help to improve insulin sensitivity in an obese rodent model. So essentially what we see here in part A, they looked at the epidemial fat tissue, and then in part B, they looked at the inguinal fat tissue. And as you can see, specifically with the TNF-A gene, you can see that the gene expression within group 5, which is again the one we want to focus on, that's that purple group, which is the mice that were treated with the tFNA resveratrol combination. The gene expression is much lower, showing that there were much less of the M1 macrophages, which are the ones that promote insulin resistance, and more of the M2 macrophages, which is what you want to see, and those ones promote insulin sensitivity. So then moving on, similar to the last slide of results, however, this one is talking about insulin resistance in the liver and the skeletal muscle rather than the adipose tissue. And so obviously, as we know, the liver is another very important tissue that is mainly targeted by insulin. Liver macrophages clearly do contribute to the production of the inflammatory mediators that promote insulin resistance. And so the high-fat diet induced obesity increases inflammatory responses in the liver, and treatments that impair these inflammatory responses show attenuation of hepatic insulin resistance. So basically in the study, what they wanted to do was they wanted to explore the effect of the tFNA RSV on hepatic insulin resistance in the liver. And so in order to do that, they took the glycogen levels in the liver, and they were assessed using PAS staining. And they didn't see any obvious differences in glycogen levels between the high-fat diet group and the RSV group. However, they did see that the tFNA RSV treated high-fat diet mice had more glycogen accumulation in the liver than just the group that was only fed the high-fat diet without any treatment. And and this group of the tFNA RSV treated mice showed much more normal glycogen levels, much more comparable to a normal mouse. So basically what this suggests is that this tFNA RSV helps to improve insulin sensitivity in the liver, and it helped to reduce insulin resistance that is induced by obesity. And so in general, again, we see this lesser gene expression from group 5, showing that there was a switch in the macrophage polarity from M1 to M2 phenotype. And then also as well for the accumulation of macrophages in the skeletal muscle, the macrophage polarity was also switched in the skeletal muscle, and this helped with overall health of both the liver and the skeletal muscle. So that was the last slide of results, and moving on to the conclusion. So I thought this was a good figure that kind of sums everything up from the study and just generally shows what was concluded. So on the right side of this figure, you can see this represents the mice that were fed the high-fat diet, but also given the tFNA RSV treatment, whereas the left side represents the mice that were fed the high-fat diet, but did not receive this treatment. And as you can see on the right, because of this treatment, there's a reduction in the size of the adipose tissue, the liver is much more healthy, and the muscles, skeletal muscles are also much more healthy, and there's overall more insulin sensitivity in the cells. And then you can also see there is promotion of the M2 phenotype of the macrophage, and there is less T cells that promote inflammation, and there is more T cells that inhibit inflammation. Whereas on the left side, without the treatment, there's much more fatty adipose tissue, the liver is unhealthy, the muscles are unhealthy, and overall the cells are much more insulin resistant, which is not a good thing. And so basically this is a good comparison showing that when given the treatment of these tetrahedral framework nucleic acids incorporated with the resveratrol, these mice benefited a lot, and their overall health and weight also benefited. So that was the main conclusion of the study. So just some general take-home points that I want to go over. So first of all, one of the basic things that the study helped to prove was that these tFNA's, they do have superior drug loading performance. They're very good at being incorporated with the drug and effectively carrying that drug into the body and delivering it and making the drug much more effective than it would normally be without the tFNA's present. As far as specific to the tFNA RSV combination, they did find that this treatment, it helped to improve insulin sensitivity in the high-fat diet-fed mice. It helped to promote anti-inflammatory T cells, and as well as inhibit the pro-inflammatory T cells. It suppressed the pro-inflammatory cytokines that are normally secreted by the M1 macrophages. And in turn, it switched the macrophage polarity from M1 to M2 so as there to be less pro-inflammatory cytokines in the body. And so essentially, in conclusion, the hypothesis of the study was supported by their results. They did see a huge benefit and much or a lot significantly decreased insulin resistance in the mice that were fed with this or that were treated with this tFNA RSV molecule. And so they saw what they wanted to see, and essentially this is really good news. And they did this in a mouse model, but they hope to maybe one day transfer this to human models in helping to treat things such as type 2 diabetes and other conditions that are related to and tie in with insulin resistance in general. And so, yeah, they would hopefully like to see tFNA's eventually down the line being used in humans to better deliver drugs and treat things such as insulin resistance. And there's my work cited. I will go ahead and stop the share. And thank you so much for listening to my presentation on insulin resistance and tetrahedral framework nucleic acids. Thank you.